So now that we've got all of that undone, our first, our next process is to talk about mitosis. Now, mitosis is by far the more basic version of cell division, and we start with this phase called interphase. So this is the base phase of the cell, and it's basically where the cell is growing, DNA replication has just occurred, and we have prepared for cell division. So the division step hasn't actually started yet. And then our first real step in the cell division process is that the chromosomes will start to condense down into the normal chromosome structure that we looked at before. Prior to that, they're not actually always in this X shape. They're kind of just floating around in the cell as these strings called, well, they're still chromosomes, but like they're still like floating around in the nucleus of the cell. Then, in prophase, the chromosomes will condense into the X shape and start to pair up with their sister chromatids. So we've got two identical chromosomes, four sister chromatids here. When the chromosomes are actually condensed like this, it's important to note the DNA can no longer be accessed. The telomeres have like zipped them all up, and the only thing that can happen from this point forward is cell division. So the DNA kind of goes into a more unwound state where it can be, oh, right here, never mind. And then the other thing that happens in prophase, along with the chromosomes condensing, is that the nuclear envelope, so the membrane of the nucleus, starts to degrade. And so the centrioles in the centrosomes right here will start to form spindle fibers. And we'll see that a lot later on in the latter steps. We then move on to metaphase. So this is where the spindle fibers coming from the centrosomes at the poles of the cell, which will hook onto each of the condensed chromosomes as they line up at the equator. So basically they hook up onto the chromosomes and pull them so that all of the chromosomes in the cell are lined up with the center of the cell. Think about it as metaphase, middle. They line up at the middle of the cell. Then what happens is in anaphase, the chromosomes which have lined up at the middle get pulled apart by the centromere. So that each of the sister chromatids gets pulled apart. And then in telophase and cytokinesis, all of the sister chromatids will reach the poles of the cell and they will start to form a nuclear envelope. So you end up with two separate nucleuses sharing one body of cytoplasm. And then in cytokinesis, the cytoplasm will split into two, and then the cellular membrane will form, making two identical daughter cells, which are diploid. That means that they've got two copies of each of the chromosomes. Right there. So, the more complicated step is our meiosis. And oops, sorry. This is our process of creating haploid daughter cells for the gametes. Again, we're starting with the same process in the sense that it follows interphase, prophase, metaphase, anaphase. But instead of having one round of that, you end up with two rounds of cell division. So in interphase, we start off with the chromosomes and the DNA being copied. This is a 2N cell as well, so we've got two copies of the same chromosome. In prophase 1, these chromosomes will pair up essentially. So you've still got two chromosomes, but they've doubled, so you've got four sister chromatids in total. Metaphase then starts to occur, so the spindle fibers from the centrosome start to anchor onto the chromosomes and line them up at the equator. But instead of the chromosomes forming one underneath each other, homologous chromosomes, so chromosomes that are of the same size, will line up next to each other, and anaphase will pull apart an entire chromosome to the pole of the cell. One thing that's important to note here is that after metaphase 1, there is this process that occurs called crossing over. Oh, what is happening to the slides? crossing over, which is basically where homologous chromosomes, so these are identical chromosomes that have been replicated. One is from the parent's side. So, sorry, that didn't make sense. One is from the father's side, one is from the mother's side, and their ends will start to overlap with each other and exchange sections of DNA. So this leg that is overlapping here, the overlapping section of the blue will belong now to the red chromosome, and the overlapping section of the red will now belong to the blue chromosome. These form something called recombinant chromatids, where basically the two copies of DNA have a combination of both the mother's DNA and the father's DNA on both chromatids. This is an adaptation that we've had that helps to improve our levels of genetic diversity because it shuffles the genes between the parents a lot more compared to if there wasn't any crossing over. The second thing that sort of happens in this section is independent or random fertilization, where each of these chromatids, whether it be a recombinant chromatid, one that has exchanged DNA with the other parent, or a non-recombinant chromatid, one that hasn't done the whole exchange, there's equal chance of these guys being fertilized. One isn't prioritized over the other. So that also helps to increase the level of genetic diversity in the gametes as well. So once crossing over has occurred and the chromosomes have shared genes with each other, anaphase starts to occur where the homologous chromosomes start to be separated to the pole of the cell. And then finally, the nucleus splits and the cytoplasm splits and the cellular membranes form to form two still diploid daughter cells. However, it doesn't stop there. We then go on to prophase 2. So we end up with two daughter cells, each with one version of the homologous chromosome in them. Metaphase 2 is where chromosomes will line up at the equator again and the spindle fibers will start to form. Then anaphase will pull each of these chromatids to this to the pole of the cell. So instead of having two copies of the chromosome like a normal mitotic cell division would have, this time we're only having one copy. So instead of 46, you only get 23 in the final one. Sister chromatids are then pulled apart. Telophase and cytokinesis occurs where the cytoplasm splits and the membranes formed. And we end up with 4 granddaughter cells that are all genetically unique. You can see, imagine if the darker one is the mother's DNA and the lighter one is the father's DNA. You've got one haploid daughter cell with entirety of mother's DNA, 2 which have combinations of both, and 1 with only the father's DNA. Each of them only having one copy of the chromosome, so it's a haploid or N cell instead of a 2N cell. Sorry, we went through that quite fast, but so much to go through with you guys. So we've already talked a little bit about crossing over, and finally, metaphase 2. At the end of meiosis, we end up with 4 granddaughter cells. However, there are 2 types of meiosis that can occur. Because at the end of the day, meiosis is making sex cells, right? So either our sperm cells or our egg cells. So there is slight variations in the production of sperm cells through spermatogenesis and egg cells or oogenesis, which is what we're going to be talking about now. Just checking on the time so that we're okay. So our first type of cell division is spermatogenesis. This is almost word for word recollection of the meiosis process. Our germline cells, which is our spermatogonium, or the original the origin point of spermatogenesis, will condense all of its chromosomes and copy them all so that they form a primary spermatocyte. This is where meiosis 1 happens and creates secondary spermatocytes. So instead of having 2 copies of every single chromosome, you end up with 1 copy of every single chromosome. Then meiosis 2 will occur and split all of those chromosomes up into individual chromatids, forming 4 identical sperm cells. Well, 4 sperm cells of the same size. They are all genetically different, though. And in terms of the timeline of this happening, after puberty, the spermatogonium forms the diploid primary spermatocyte through mitosis, which then undergoes meiosis 1 to form haploid secondary spermatocytes, and then meiosis 2 to form haploid spermatids. So these are our end cells at the very end. We then see that spermatogenesis is very closely following meiosis, forming two distinct, however, size identical sperm cells. These are then what fertilize the egg cell during fertilization and end up forming a zygote or fetus. Oogenesis, or the production of egg cells, is actually a lot more complicated. So before birth, all of the all of females will have a set number of eggs that are each 2n, called oogonium cells. And then through mitosis, all of their DNA will split into primary oocytes. However, this stays the same in prophase 1 until puberty hits and before the meiosis step can actually occur. So after puberty, you end up with meiosis 1 happening, which when it splits, so wait, let me go back. In meiosis 1, for spermatogenesis, we had two identical spermatocytes, right, in terms of size and structure. For meiosis, instead of them being identical in terms of size and structure, one of them will be bigger and one of them will be smaller. This is because at the end of oogenesis, we're not ending up with 4 viable egg cells. It's just not going to work that way. An egg cell is one of the biggest cells that you can have in your in the human body. And the fact that it becomes the basis for the zygote or the baby that comes afterwards means that it has to have a lot of cell mass for nutrients so that the organs can start developing, and a much larger, a much larger cell mass full of nutrients and things that allow a cell or a baby to start developing. So instead of meiosis 1 resulting in 2 identical oocytes, we get 1 secondary oocyte and 1 polar body. This is straight after puberty. Then, after ovulation, meiosis 2 starts to occur, and you end up with. Oh, hold on. Then, after ovulation, hmm, hold on. Mitosis occurs before birth, then meiosis continues after, and you get a secondary oocyte and a polar body. After ovulation, sperm will start to enter the cell. It undergoes the second wave of meiosis, fertilizes, and you end up with a fertilized egg and a polar body. Oh, oh my god, what is happening? So, with actually, let me go back to this. With oogenesis, instead of forming 4 viable gametes or 4 viable egg cells, you end up with 1 viable egg cell that has taken most of the mass of all of the others, and 3 polar bodies which eventually have to be discarded. One of the major differences between oogenesis and spermatogenesis is that spermatogenesis forms 4 the sperm cells of identical size and structure with different genetics, whereas oogenesis forms 1 egg cell with unique size and structure and 3 smaller polar bodies, which then have to be discarded by the body. So